Hi, Millennium. So you are the first time, right? What are you doing? Where are you studying right now? So what is the course? Can you tell me? You can text actually.
I'm from Assam Don Bosco University and I'm
Okay.
doing MSc in zoology.
MSc Zoology. First year or second year? MSc Zoology. Are you in first year or second year?
Ma'am, I'm in second year.
Okay. Okay. So you must be knowing most of the things, right? We'll wait for some time. More people will join, then we'll start. Okay. Hi, Kandipora.
Hello, ma'am. Good evening.
Good evening. How are you?
I'm fine, ma'am.
So are you guys able to do the assignment question? How is it? Is it easy or is it okay, okay?
Yes, ma'am.
Okay, next. We'll wait 5 more minutes, then we'll start. So did you listen to last week lectures? Week 6, neural system. I hope you guys listened already. What sir was teaching in the last week? 5 lectures, starting from the first day to fifth day. Any idea? Anyway, I will give like the summary of each lecture slides, what sir was mainly teaching. Just go through the main topics. Then we'll try to recollect whatever he was teaching. Then we'll try to solve the questions, some demo questions, so that it will be easy for you guys to go and do the assignment. And also today I will like last week, fifth week solution they released. So we'll go through that also. I know you guys already attempted, but let's solve that also after the trial for the sixth week. Then we'll go ahead with the fifth week questions. Happy Teachers Day, by the by.
Happy Teachers Day, ma'am.
So I will give my intro again. People will join, I guess. Yeah, let them. So I am Harsha. My name is Harsha Shankar SH, and I am a PhD scholar in ISET Berhampur, a PMRF fellow. So I am basically an integrated PhD student here. So I completed my MSc here only, and I started with my PhD. Now I just started my second year of PhD. So that's about myself. Hi, Demon. Sorry if I am mispronouncing your names. Okay, forgive me. So I will share the slides. Okay, then we'll start. Hi, Vishwapragyan.
Hello.
Hello. So I think you guys are new in our interactive session. So can you please introduce? Okay, let me come back one minute. Yeah. Hi, Udit. Hello, Ramesh, Dr. Ramesh Doreswamy. So today we are going to do the live interactive session of the week 6. Since more people joined, I again give my intro, then we'll start with today's session, okay? So my name is Harsha. I am a PhD scholar in ISEL Berhampur in Department of Biological Science. Okay. So yeah, coming back to the slides. My slides are visible, right?
Yes, ma'am.
Okay. So basically, week 6 was continuation of week 5 only. I hope you guys went through all the 5 lectures. So lecture 1 started with mainly with the topic of spatial temporal summation of signal in mesh neurons. Then in that, like Sir was mainly telling us about the how the usually neurons are propagated, like from one neuron to another, and mainly regarding the inhibitory signal and exhibitory signal. So if in the presynaptic neuron exhibitory signal or the neurotransmitter is releasing is a exhibitor kind of thing, what will happen in the postsynaptic neuron? It is kind of activating. So in the last week also we discussed how they are activating, how the activating and inhibitory like neurotransmitters are basically different. So always the excitatory neurotransmitters, what they can do, they can open more positive ion channels, right? So it will trigger again a high signal response. If it is inhibitory signal, what will they do? They can open more negative ion response, and so the function will be opposite to that of the excitatory neurotransmitter. So then so different types of neurotransmitters are there. So in like at a one point, from one neuron, excitatory is coming, and from the same adjacent neuron, inhibitory is coming. So how can we totally add up? If it is like some 15 unit and this is like 20 units, sir nicely explained that we can add up. So if the excitatory was 20 and inhibitory was 15, net effective is also plus 5, right? 20 minus 15. So it will be again excitatory, but the 20 is now reduced to 5. So such kind of changes are happening always around the synapses based on what kind of inhibitory neurotransmitter or excitatory neurotransmitters are acting. So that is basically was the first introduction or first topic in the class. Then there is a mobility in all these synapses. Like as I said now, if depending on the amount coming, how the presynaptic neuron, what is the signal coming from presynaptic neuron, and how the postsynaptic neuron is responding to the signal from the presynaptic neuron. So based on all these activity, there is a synaptic mobility. So we'll explain what is it more detailing in the coming slides. And then also he told about what is hyperexcitability. So what is excitability? As we mentioned already, right? If the presynaptic neuron is excitatory in nature, it will definitely going to, and if suppose any inhibitory signal is not acting on the postsynaptic neuron, only the excitatory neurotransmitter is acting. So what will happen? Definitely the excitatory neurotransmitters are going to continuously excitate the neuron, right? Suppose this is like normal case. In normal case, there are balance between all these scenarios. So there are some things to bring back the neurons excitability to the normal condition. Suppose in particular regions of the brain, what will happen? There is some like researchers are even ongoing to how the changes are happening, how the neurotransmitter neurons is acting differently, how they are continuously releasing the excitatory neurotransmitters. All these are under the research questions. But if suppose the presynaptic neuron is continuously releasing the excitatory neurotransmitter, what will happen? It will overexcite the postsynaptic neuron, or it will also start doing the same, right? So such a condition is called hyperexcitability. Obviously this is an abnormal condition, so definitely there are some disorders which is arise due to such condition. So one such is disease is the epilepsy. So do you know what is epilepsy? Anyone? So what is epilepsy? It's written also there. So epilepsy is basically the disorder of the brain that is characterized by repeated seizures, right? So how the seizure is happening? As I said earlier, like there is a, sorry. So how the seizure is happening? Because there is a neurotransmitter release, which is excitatory neurotransmitter release happening, and that will is continuously happening. So which is again excitating the adjacent neuron or postsynaptic neuron, and so that's why the hyperexcitability is happening. So what will happen eventually? Brain is not understanding what is happening there, so it will leads to the repeated seizures, and finally, particularly the fainting up of the person. So that is the condition characterized by epilepsy. So this was the first topic. Then in the second lecture, it was the mainly regarding the anatomy of hippocampus. So if we need to tell about the hippocampus, we know our brain. So in the first lectures, we studied like dorsal view of the body, ventral view of the body, how like how what about all the divisions that can be body can be divided, right? So if we are checking the dorsal ventral view also, if we are sectioning different part, there will be different sections, right? So there is a region adjacent to like this is total hippocampus in the brain. Actually, in the slide it was there in the start slide. So brain below it looks like basically if you're checking the whole total of the structure, how it looks like? It looks like a seahorse, right? So that's why the name, interesting name, come for this structure as hippocampus because it is looking like a seahorse. And there are different region inside based on the dorsal and ventral divisions. Like there are CA1, CA2, CA3, CO4. And also, he told about what are the majority of neurons that are present across this region. So if we are going and checking this region, we can see some interesting shaped neurons. like a pyramid. You guys know about the pyramid, right? So those neurons, majority of the neurons found in this region majorly looks like a pyramid in structure. So that's why the name comes for that neuron as pyramidal neurons. Then also there is a part, right? You can see in the figure there is a blue part, right? This blue part is the dentate gyrus. Actually, this is a very relevant area. Why? Because earlier people even don't know that there are some progenitor cells that are present in the brain. Then people found out that there is these regions. So in the dentate gyrus also is one of such region where there are some neuronal progenitor cells are there. So these cells, the cells present at this area are particularly able to produce the new progenitor cells. So this is also a area of great interest, especially for researchers. Secondly, so there are majority of pyramidal neurons present. Then secondly, after pyramidal neurons, what is the most abundant neuron present across this region? That is the interneuron. And also he told about what are the major neurotransmitters, like how what all types of neurons are present there. So these pyramidal neurons or interneurons are mainly consist of two neurotransmitters. Did anyone remember what are those two main neurotransmitters that see across this part? Mainly two neurotransmitters are there. Yeah,
GABA
yeah, right. Okay,
and
Arya.
glutamine.
Yeah, so majority it is glutamate. Then second one is GABA. And also very minute amount, one more is present. What is that?
Acetylcholine.
Yeah, very good. So these are the three neurotransmitters that we can. So which means that mostly these pyramidal neurons and interneurons are mainly glutamate, glutamic neurons or GABAergic neuron or cholinergic neuron, which it comes in a very less proportion. Okay. So that is about the anatomy part. Then coming to the next lecture, it was mainly regarding. So you understand what is dentate gyrus, what is pyramidal neuron, everything, right? Lecture 3 was mainly regarding the epilepsy and memory, right? So it was really interesting to listen to that story, right? In one of the Canadian person in 1940s, what did they do? So this guy was affected by mainly epilepsy, like repeated seizures. The seizures was continuously occurring. So doctors have an idea that how the seizures, these things are happening, which part of the brain is causing a problem, or where these neurotransmitters are causing a problem. So people know that here in the slide, I know the clarity is very less, but you guys always already seen the slide. So you guys can see the pink shaped, seahorse shaped hippocampus here, right? So they know that this hippocampus part is the whenever some neurotransmitters, like there are some hyper excitable neuro transmitter problems are happening, or hyper excitable neurons are there. That's why these repeated seizures are coming, and as a result epilepsy is causing. So what did the doctor do? So they have decided, like discussed with the patient, and they have decided to remove both of the seahorse structure, right? What was the interesting result? Yeah, like the surgery was medically successful, because after that he never got the epilepsy back, which means epilepsy is rid of epilepsy successfully. But unpredictably, or what, it's like a, we don't know, accidentally something else they observed, which was the changing, one of the medical changing observation for the neuroscience researchers. So what did they do? They observed that this person is affecting by memory loss. He is unable to recollect the memories. So then they found out that this structure or seahorse shaped structure is also is the memory center of the brain. So always question comes, what is the memory center or learning and memory center? So please be remembered to write it is hippocampus. Okay. So mainly that was the hippocampal removal experiment. Accidentally that guy got lost all the memories. So what is memory? Then how memories are formed. Okay. So there are like short-term memory and long-term memory and like long-term potentiation, long-term depression. So all these things, the later slides, he also told about what is long-term potentiation and what is long-term depression, all those things. So here still there are so many questions acting. That's why even this sir was basically addressing on the main focus question. Even this is a question that how the basically hippocampus is able to do this, or how the memories are stored in the hippocampal region, how this is happening. It's like there is no complete answer to any of these things. So like there are always some assumption, like for epilepsy people assume that maybe hyperexcitability is due to there are some maybe the genetic level changes, or there are extra some genetic level modification present across those neurons which makes them hyperexcitable. So all these like such like some kind of weight ideas is there which is not like completely understood. Okay, so that was about the epilepsy and memory. Then coming to the fourth lecture, it was long-term potentiation. So before saying what is long-term potentiation, we should remember of a person who is Donald Hebb. Okay, what did this guy found out? So he said, whenever there is a axon of a cell A, okay, a adjacent axon of a neuron, first neuron is A. And this A is always exciting the adjacent neuron, which is B, suppose. Okay, so A is repeatedly exciting the B. Okay. So now what will happen? So he said that there will be some metabolic changes associated with this process, like A's release of neurotransmitters and B acceptance of there is some metabolic changes happening, and as a result of continuous repeat activity of A, which is making B also like high firing cells, high firing neuron, which means A is basically due to A is repeatedly releasing the neurotransmitters, which is increasing the enhancing the activity of B. So that is called as Donald Hebb mode. So what is relation with long-term potentiation? So it is just think of in this way that so since A is firing continuously, so B is exciting, right? So due to some function of the presynaptic neuron, postsynaptic neuron is also getting excited. So such type of things can be regarded as long-term potentiation, where the persistent strengthening of a synaptic connection is there. Due to A's activity, B's activity is strengthened. So it is always based on this Hebbian principle. So as a result, the cells that fire together will wire together. And also, sir explained nicely, like, so suppose if A is like giving an instance, what was the instance? We are focusing on something very, like, very concentratingly. Suppose take any of your interesting activity. Okay, I will, okay, suppose you are listening to a music and you guys are still listening to a music, still listening to a music. Okay, suppose the music is not there now, still, but you are getting a perception that that is still going on, which means A's activity is not happening. Still, you are getting a perception that the B is that activity is still going on. So such kind of things are called, which means due to the activity of A, B is potentiated or B is changed. So that's why this is termed as long-term potentiation. So then there is one more type, which is the long-term depression, where the lecture started with basically telling about the emotion and fear memory. Suppose you are seeing a snake. I don't know how many of you are actually scared of snakes, but yeah, whenever you are finding a snake, you don't know. Suddenly you are realizing that, oh, it's like a snake, and something is coming into your mind. You are making realize that, oh, some changes are happening, right? It's it's same regarding the emotional and fear memories. So whenever like if some before like some years before, if suppose your grandma told you some horror story, and whenever you are in such a condition where you are able to recollect to the backstory, again the same thing is happening. Some sudden emotion or fear memory is coming, right? So, which is the center that regulating emotion and fear in the brain? In this lecture, he actually told what is that structure. It's not hippocampus. Structure in the brain that regulate emotion and fear memory. Which is that?
Is it amygdala?
Yeah, correct. It is amygdala. So amygdala is also, I think in some of the question it is coming. So we can show that figure also. So amygdala is the center of emotion and fear memory. So what is coming in long-term depression, and he also told very nicely that, like suppose if you are in a 5-year-old, okay, definitely your 5-year-old and 10-year-old, if you're comparing to with 20-year-old, 30-year-old, 40-year-old, 50-year-old, as the age progresses, there is definitely difference in the information storage due to why information storage is changing due to the changes in the microanatomy. If we are dissecting and checking a 5-year-old same person with 10-year-old person with 20-year-old, like the same person, we are doing this experiment in like this much course of time, we can see the changes inside the microscopic level changes inside the brain. So he also explained that thing. Then we'll give an like basic introduction about what is LTD or long term depression. What is long term depression? It is opposite to long term potentiation, right? Earlier, what was in long term potentiation? What is happening? A is exciting peace activity. So here, what will happen? LTD is expressed by long-lasting decrease in the efficiency of synaptic transmission. Due to the X activities, these synaptic transmission is get delayed. So such condition is characterized by the long-term depression. And still there are like not complete answer why it's happening. But there is one big idea that it's due to the activity of change in the activity of some particular type of receptors. This AMPARS. Anyone know what is AMPARS? You don't need to remember the full form that I have written in the slide. But what is AMBUS? Like what kind of receptors are there? We told already now what are the major types of neuron present across these parts. Since we are talking about memory, it's mainly regarding, I am telling about describing to hippocampus. So in hippocampus, what all types of majority of neurotransmitters are there?
Glutamate and GABA.
Yeah, so AMPs are basically glutamate receptors. Okay, so what will they do in long-term potentiation? Basically, the AMPAs will be due to the inflow of some change in the ions. AMPAs will be more present, and AMPAs taking continuously glutamate. So in long-term depression, it will be opposite, right? Which means that AMPAs will be much activity will be much blocked somehow, and they are not continuously taking the neurotransmitters. This is just for an add-on information I gave it here. Okay. uh, so we'll start with the question. I hope you guys understand. I was just rushing through the basic summary of each lectures. Okay. So let us come back to the questions. Sorry. Yeah. Which of these brain region is associated with memory and learning? A hypothalamus. Oh, okay, okay. So I will tell you the rule. Okay, since how many people are here? 1, 2, 3, 4, 5, 6, 7, 8. Okay. So the rule is I will read the questions. You guys can. I will check. Okay, how many like answers I am getting. So you guys start typing your answers in the chat box. I will wait for some time. Then we'll after some time we'll start discussing the answer. Okay. Is it fine with everyone? I hope it is fine with everyone. Okay, so first question. Which of these brain region is associated with memory and learning? Option A, hypothalamus. Option B, amygdala. Option C, visual cortex. And option D, hippocampus. Okay, one answer came. Others, please. That's why we rush through the summary so that we know. Yeah, Kushbu Kumari also wrote the answer. Very good. Others, please. It's easy question. That's why we started with the class. We are continuously telling about that structure again and again today. Two answers only came. I am waiting for 6 more responses. Okay. 3 people wrote. Come on. Okay, four. 4 more. Okay, five. 3 more. Three more. Okay, they are not interested to say the answer, I guess. Okay, then we'll discuss. So, what was the question? The brain region associated with memory and learning, right? So, what is the center? Let's see. Answer is B. You already told always the memory and learning. That's why we told about that experiment also. The person accidentally, when they removed both hippocampus from the brain, what did they observe? He lost the memory, which means like hippocampus is always the memory or learning center. So correct answer is D. If any doubt is there, you guys can ask, okay? And amygdala, yeah, we'll go through it. I know you if there is any like doubt in option also, you guys can ask, okay? So next question. Hmm. This is from the last week part actually. During depolarization, which ions move inside the cell from outside? Option A, sodium ions. Option B, potassium. Option C, both A and B, both. Option D, none of sodium or potassium. Question is during depolarization, which ions move inside? We discussed actually this a lot in the last week, and this was the basic starting of how the neuron activity is going on. So just recollect whatever we learned, what is resting membrane potential, what is depolarization, what ions are triggering the process of depolarization, then what is hyperpolarization, like what all receptors are playing a major role there, all these things. So let us see what are the answers. Yeah, yeah, write it. Okay, you guys already write it. Oh. Option A, option A, option A, option A, option A. Okay, correct. So you guys are very smart people. You guys are remembering whatever we learned in the earlier class. Yeah, this figure you remember, right? In the last lecture, Sir was drawing this on the board. So here you can see that whenever depolarization is happening, whenever the action potential is attained by a neuron, then the inflow of sodium will happen, which will make the positive inside the membrane positive combined with outside. So in order to, so this is mainly happen to the opening of sodium. What type of ion channel?
Voltage gated.
Sodium voltage gated ion channels. Then cells are understanding that oh, there are some disturbance happen. So in order to bring back the potassium voltage gated ion channels will open, which will leads to the outflow of potassium, right? So potassium will start going out, which will make again the inside the membrane negative compared to the outside. But this will sometimes keeps continuously going, which will make the inside the membrane more negative. Then in order to bring it back again, the sodium potassium pump will open due to the outflow of 3 sodium outward and inflow of 2 potassium inward. What will happen? So cells will be finally maintained at the resting membrane potential. So that was the whole process. So you guys know this. Let's see the next question. So I know this is like we never discussed this. So you guys can discuss, just guess, okay? Optimum sodium ion concentration for extracellular fluid is option A 138 to 146 micromole per liter, option B 138 to 146 millimole per liter, option C 230 to 500 micromole per liter, and option D 230 to 500 millimole per liter. Any idea? Just guess it. It's fine. Okay, Arya already texted. We'll wait for others to also text. Just guess it, it's fine. Okay, Kushbu is saying option A. Okay, let's see. Okay, everyone is saying option A. It's a guess, it's fine. Let's see, okay, what is the correct answer? So basically, this is about the whole sodium concentration. The mean body content of sodium in an adult mean is 92 gram, okay? And half, which means among the 92 gram, almost 46 gram is located in the extracellular fluid. So it must be at a concentration of 135 to 145. So here in the option, you can relax. Just check the unit and the number. This is millimole per liter, right? So millimole per liter and 135 to 145. So the correct option is answer B, right? And intracellular, it will be 10 millimole per liter, which means that majority of sodium are present in extracellular fluid only. That's why it is present outside the membrane very large in number, and whenever there is a opening of voltage gated sodium ion happens, it will rush inside, right? So just keep in mind that this is about the concentration. Okay, next question is, which of the following cell types form the population of the dorsal pathway of the spinal cord? This also we discussed last week. Dorsal pathway, ventral pathway, or efferent pathway or afferent pathway, and what are the neurons connecting it? All these things. Option A, motor neuron. Option B, interneuron. Option C, sensory neuron. Option D, all of the above. You guys can start answering. Okay, sensory. One answer is there. Option A, option B, it's fine. I actually forgot to add the option A, B, C, D. Okay, Candy Bora also texted. Come on, others also, please. Four people gave answer. Is anyone else? Come on. Okay. So answer is C only, sensory neuron. Let's see. Okay. So this was the pathway. I hope you guys remember. So what is afferent pathway basically? That will connect information from the sensory organ to the central nervous system, which can be brain or spinal cord. So that pathway is called afferent pathway. Then what will happen through the efferent pathway? So the red colored neuron, which is called motor neuron. So here these blue colored neurons are sensory neuron. So sensory neurons are involved in the afferent pathway, or the afferent pathway is one more name is there, that is the dorsal pathway. Okay. Then next question. I can move, right? We already discussed this part. AMPA is A, glutamate receptor, GABAergic receptor, cholinergic neuron, or none of the above. Option A, option B, option C, option D. What is the correct answer? Okay, Arya is saying glutamate. Okay, option A. Kushbu is saying option A. Damon is saying option A. Everyone is saying option. Okay, I am really happy that you guys are listening to the whatever like we are discussing here. Very good. So yeah, we already discussed, right? AMPA is a glutamate receptor. So I don't think I need to give more explanation. Then next question. Okay. So I have given this also. Basically, there are two types of receptors: ion channel associated receptors and G protein coupled receptors. So this is basically what all receptors are present across the glutamate, like what all are the glutamate receptors. So in inotropic, there are different kinds which consist of NMDA, AMPA, and these are the two main. Then there is also kinase also. In metabotropic, there is group 1, group 2, group 3. So all these are either excitatory or inhibitory. So mainly inotropic are always excitatory and some metabotropic are inhibitory. So this is majorly about what all are the receptors. Then next question. Okay. Which of the following cell type exists in ascending pathway of spinal cord? Look at the importance of this type of question. Again, they are asking either ascending, afferent, afferent. So they are just changing the words and again asking the same question. Okay, Arya started. Sensory. Everyone else, please. Okay, Shaila is saying C. Okay, Candy Bora is also saying option C. Come on. Three people answered. More to go. Just guess the answer. No need to guess. I know you all know the answer. Okay, then let's discuss. Yeah, the option is C only, right? Why? Ascending pathway means it is collecting information from the sensory organ and delivering it to the spinal cord or the brain, which means that the ascending pathway or afferent pathway, whatever the pathway, the neurons involved are always sensory. And if it is delivering the information from the CNS to the PNS or to the sensory organ, that response pathway consists of efferent pathway, and efferent pathway or descending pathway consists of motor neurons. Okay. Now, next question. Which of the following cell types are part of the ventral pathway of the spinal cord? Again, motor neuron, interneuron, sensory neuron, Purkinje fibers. Option is same, just they are checking, you know, what is the different name of the pathways. Okay, two answers already came. Three answers came. Four answers came. One, two, three, four, five. Okay, five answers. No, four only came. No, no, six answers came. Okay. So let's see the answer. Yeah, it's right only because we already showed that figure in the earlier slide, right? Do I need to go back? This one. So what is the, we just discussed, right? In the efferent pathway or the descending pathway, or what is the another name that is provided in the question, ventral pathway. So ventral pathway descending pathway or efferent pathway is consisting of what kind of neuron? Motor neurons. So correct answer is A. In epilepsy, neurons become option A non-excitable, option B Schwann cell, option C hyperexcitable, option D none of the above. Okay, let's see. Did you guys already started? Yeah. Okay. Kushbu, Arya, Candibora, Demon. Four answers came. Five answers came. Yeah, we already discussed, right? What is happening in epilepsy? Due to some problem, the neurotransmitter is continuously releasing from the one neuron, which makes the other neuron hyperexcited, right? So that state is called as hyperexcitable. So already gave the introduction in the intro part of this session. So I'm not going back. If I want to go back, just please let me know, okay? First model of learning was given by Donald Hebb, Maynard Das, James J. Hickman, Rosalind Franklin. Okay. Yeah, three answers came. Four answers came. Correct enough? Yeah, four answers. So everyone is saying option A. Donald Hebb, right? Okay, let's see the answer, okay? Yes, it's correct because we already discussed. You remember the photo of the person also. He proposed when A is continuously triggering, we will get excitable. So correct option is Donald Hebb. Next question is part of the brain associated with fear or emotion is option A hypothalamus, option B amygdala, option C visual cortex, option D hippocampus. Okay. Answers started coming. Okay. Everyone, please. We already discussed this. I guess almost all people answered. Let's see. Okay. Yeah, we already discussed, right? This is the structure of amygdala, this yellow colored one. Right? It's near to the hippocampus. You can see a seahorse shaped structure, and near to it, there is a center of amygdala. So, always regarding the fear and emotion, everything is controlled by amygdala, where the long term depreciation part also comes to play a major role. Next question is okay. So, first week question done. So, it can really go if someone not attempted any of the assignment question, please go and complete it today itself so that I guess you got an idea about what all things. And if you have any doubt also, please feel free to ask. So then actually question last week solution is released. I guess you guys already went through that, but we'll discuss that. Okay. Last week like question discussion. So we'll start with that. Is the lecture week 6 part is completely clear about the mainly about hippocampus, Faltar memory, and some interesting experiments, all those things. Okay, I hope there is no one raising hand or asking question. Yeah, so I am going ahead to the last week's question discussion. Question was energy of UV light is option A lower than visible light, option B higher than visible light, option C equal to visible light. So we just want to relate with only one electromagnetic spectrum, that is visible light and UV light. So which is higher? Just drop the answers, then we'll discuss, okay? Start, yeah. Arya started. Okay. I know you already attempted this question, but let's discuss. It's fun to discuss it together, right? I guess three people answered. Others, please wait for some more time. Then we'll start discussion. Only three people answered. Okay, for energy of UV light is compared to visible light, UV light energy is higher or lower or equal. Okay, then let's discuss. For knowing this thing, we just need to know about only one equation, right? Energy is equal to H into nu, which means E is always proportional to nu. E is energy, nu is frequency, H is Planck's constant, which means energy is always proportional to frequency. And we know that nu is equal to c by lambda, where c is equal to velocity of light and lambda is the wavelength, right? Which means energy is proportional, directly proportional to frequency or inversely proportional to the wavelength. So as higher the wavelength, lower the energy. As higher the frequency, higher will be the energy. So now we just need to find out which wavelength is more, UV rays or visible rays, right? So yeah, you can see here in the figure, right? Here the frequency is decreasing and wavelength is increasing. So frequency is decreasing. Here you can see where ultraviolet is coming and where visible rays is coming just below that, which means visible rays have visible rays have just tell me, visible rays have lower frequency or higher frequency compared to ultraviolet,
Lower frequency.
which means their energy will be,
Lesser than UV.
yeah. The question is energy of UV light is. So definitely energy of UV light is higher than the visible light. Option B, right? So again, same type of question. Wavelength of visible light is dash and the frequency is dash than the UV light. Same question. You just need to just change, just they are checking that your concept is very rigid regarding this. Lower, lower, option B, lower, higher, option C, higher, lower, option D, higher, lower. Just fill in the blanks with the correct option. So you just need to tell me what option is correct. Option A, option B, option C, or option D. Priya, Archana, Arya, Kushbu, Shaila, Narendra, Demon. So everyone is saying option C. Let's see. Option C, let's see. So, yeah, again the same question only. We know compared to the ultraviolet light, visible light have lesser frequency, right? And higher wavelength. So definitely higher wavelength and lower frequency. Option C. Yes. Now next question. Which of the following is a not a polymer? DNA, dextran, RNA, sodium. Everyone. Yeah. Okay, then let's go. Yeah. All answers are option D. Okay. Then I will ask again question. Okay. DNA is the polymer of what component? Monomers of DNA are called. You can either text or you can unmute and tell me the answer.
Nucleotides.
Okay. Option B, dextran. Is it a polymer?
It's a type of sugar.
Yeah. So it's a polymer of glucose, right? Then C, RNA. RNA is also polymer of what?
Nucleotides.
Nucleotides consist of what all components?
Sugar, phosphate, and nitrogen base.
Yes, right. And only sodium is there. So definitely answer is option D. Sodium is what? Element,
The
right?
platinum.
If it's a charge, it is an ion. Definitely in the neutral state, it's an element. Which of the following are found in DNA? Adenine, guanine, thymine, cytosine. Option B, adenine, guanidine. Option thymide or cytosine. Adenine, guanine, uracil, cytosine. Adenine, guanidine, uracil, cytosine. Just check with the spelling and also be careful to take the option. Option A, option B, option C, or option D. Yeah, answers are already there. Everyone is so confident. Is anyone else? Yeah, let's see. Everyone answer, then we'll discuss. Did everyone answered? I guess mostly we got answers. Yeah. So correct option is, answer A. You guys are correct. So we already discussed DNA or RNA are polynucleotides, which is made up of nucleotides, and nucleotides consist of sugars, phosphates, and sugar, phosphate, and nitrogen bases.
Nitrogen
You're
base.
right. Yeah. And attached to sugar, there are four types of bases present in DNA. That is adenine, guanine, thymine, and cytosine. If it's RNA, what is will be different?
Thymine will be replaced by uracil.
Sorry.
Thymine will be replaced by uracil.
Yeah, correct. Thymine will be replaced by uracil. In a cell, concentration of dash is higher outside the cell and concentration of dash is higher inside the cell. Option A, sodium minus potassium minus. Option B, sodium plus potassium plus. Option C, potassium plus sodium plus. Option D, potassium minus. Option sodium minus. You guys should know first the charge of these ions, then should know which will be abundant in outside the cell compared to inside the cell. Everyone is saying option B. Is there any different options? Option B, option B, option B. How many people answered? I guess 6. Yeah, 6. Now there are almost 10 people. 4 more people are not answering. Okay, let us see. We already discussed so many times, right? Obviously, correct answer is B. Because which are the ions that are almost present outside the cell in higher amount? That will be sodium and chloride. They are always higher outside, right? Sodium is plus charge and chlorine is fluoride, which means Cl minus. They are abundantly present outside the cell, right? And potassium concentration is greater always inside the cell. That's how, like depolarization, back to depolarization, they are bringing back the cell from depolarization to the normal state. So what is this amount? Potassium concentration is 5 millimolar outside and 140 millimolar inside, which means potassium is always higher concentration inside the cell. But sodium is 12 millimolar inside and 140 millimolar outside. So you can see the difference between even the about the concentration inside and outside. So correct option is answer B. Resting membrane potential of a neuron is dash. The negative sign indicates that there are more dash outside the cell than those inside the cell. Option A minus 70 positive ions. Option B minus 70 negative ions. Option C minus 10 positive ion. Option D minus 10 negative ion. Let me see answers. Okay. Started with Arya, I guess. Yeah. Arya, Shaila, Priya. Okay. Two options are there. Option A or option B. Mostly people are saying option A. Some people are saying option B also. Okay, let's see. Let everyone answer, then we'll discuss. which means if it's only option is A or B, you guys are sure that resting membrane potential is minus 70, not minus 10 millivolt, right? Only confusion is negative sign indicates that there are more dash outside the cell than dash inside the cell. Question is, what is more present in outside the cell? If it is negative, to make it inside the membrane to become negative, what should be higher in number inside the cell? Cations or anions?
Anions.
Right? It is cation basically, but the concentration there is different. That's how it's maintained. Yeah, but inside of the net charge should be higher inside the cell is negative due to the presence of, right. So in the outside the cell, which will be more positive ions or negative ions? Okay, let's see the answer. Yes, correct. Is there anyone having doubt? Then we'll discuss it more. It's always resting potential is always near to minus 60. So it's like majority it is minus 70 millivolt. That you guys don't have any doubt. The only doubt is negative sign indicates that there are more dash outside the cell than those inside the cell. So the confusion is coming. Maybe you guys don't listen to what is outside and what is inside. So yeah, definitely in outside the cell, which will be more, positive ions will be more. Like sodium ions will be more, right? So option A is the correct answer. Can I go to next question? Is it okay? If anyone having any doubt, we'll discuss. You can type yes or no. If it says I will go to next question. If it's no, we'll discuss it more. Yes. Okay. Next question. A redistribution in the electric charge of a cell occurs. Okay. Electric charge got redistributed. It leads to a less negative charge inside the cell than outside. It is called less negative charge inside the cell than outside. Usually inside the cell is negative. Now after the redistribution, what happened? Inside the cell become less negative than outside. That process is option A, hyperpolarization, option B, polar peaking, option C, depolarization, and option D, sinoatrial faulting. Okay, there is so many options. I didn't see option C, option C. Okay. Is everyone is agreeing to option C or anyone have different opinion? Option C again. Okay. Option C. Everyone is saying option C. What is hyperpolarization? Polar peaking and sinoatrial faulting, leave it, okay? We didn't learn about it. We learned two terms in that, hyperpolarization and depolarization. What is hyperpolarization? Arya was about to speak, I guess.
The inside becomes more negative.
Yeah. So its question is opposite, less negative, or maybe positive, close to positive. So that process is called as depolarization, right? Depolarization is happening due to the opening of what channel?
Sodium.
Okay, what kind of sodium channels? There are different types.
Voltageated.
Okay. Again, this is like very basic part. So we make sure that we are like confident in the depolarization, hyperpolarization, about the ion channels, about the type of neurotransmitters, what all types are there, excitatory, inhibitory, and the different types of neurotransmitters, the names comes from how, and about the synapse, synaptic junction, synaptic cleft. So we make sure that the basics we know. Okay. So the correct option is, let's see, answer C, depolarization, right? Depolarization have one more name. What is that? It's written, if the opposite is hyperpolarization, then therefore it is also called as hypopolarization, right? So it will make finally the inside the cell less negative charge or more positive. So that is about that question. Then going to the next question. Axon hillock is an dash structure and synapse is an dash structure. Option A, interneuronal. So basically that option is saying both are interneuronal. Option B is saying axon hillock is interneuronal but synapse is intraneuronal. And option C is saying axon hillock is intraneuronal but synapse is interneuronal. And option D is saying axon hillock is intraneuronal but synapse is intraneuronal. Both are intraneuronal. So this option A, option B, option C, or option D? So I guess question answers are there. Option C, option C. Okay. 4 people answered. 5 people answered. 6 people answered. Okay. Is there any question? I didn't understand why you raised the hand. If you have question, you can unmute and ask. Or is it a support to the current ongoing options? Okay. It's to as a support. Okay. So everyone is saying option C. Okay. Why? Let's see. What is axon hillock? And we also in the earlier one of the discussion, we also told one interesting fact about axon hillock. What was that? Some generating point of something.
Generation of action potential.
So action potentials is always generating from axon hillock, right? Yeah. Here the answer is, you guys are right. Option C. Why? Because we know axon hillock is present inside a neuron. We know it starts with the different branches of dendrons, which are called dendrites. Then dendron is there, which is connecting to cell body. Inside the cell body, there are different cell organelles, and cell body is leading to the long branch of neuron called axon. The connecting point of cell body and axon is axon hillock is there. Then synapse, where synapse comes, axon will end in some synaptic knobs, or axonite will end in synaptic knob. Then these neurons is not physically connected to the another neuron. There is a gap. That gap is called a synapse, right? That gap is called a synaptic cleft, and that process is known as synapse. So basically, that synapse junction is a not within the neuron, right? It is a space between two neurons. That's why it is interneuronal. So correct option is option C. Let's go to next question. For action potential to occur, the potential needs to shoot below minus 100 millivolt. Option A. Option B. Shoot above minus 30 millivolt. Option C. Remain between minus 60 to minus 70 millivolt. And option D. Remain at resting membrane potential. Option A, option B, option C, or option D. Arya saying option B, Kushbu saying B, Damon saying B, Priya saying B, Shaila saying option D. Okay, others. Action potential to occur. Potential needs to cross some potential. What is that potential called? The minimum potential that. So cells, every time this is happening, right? Yeah, threshold potential. So what is the threshold potential here? So answers are majority are saying option B. Okay. Is everyone answered? Anyone else have different opinion? Means I don't know. Whatever the answer you can say, it's fine. Absolutely fine. Okay, let's see the answer then. Correct answer is option B because threshold potential is above minus 30 millivolt, right? So if any cell needs to start the action potential or start the process of depolarization, what channel should open? As earlier you guys said, the voltage gated sodium channels should open. It can open only if the threshold potential attains or the potential crosses minus 30 MV. Right? So you know minus 70 and minus 60 are resting membrane only. If the cells are at resting membrane potential, it's in a peaceful state, right? So there is no chance of potential to rise. So when the potential resting membrane potential start crossing or attaining threshold potential, then it will start opening all these voltage gated sodium channels and inflow of sodium channel sodium ion through sodium channel occur, which makes inside of the cell become positive or less negative compared to the outside, which is known as depolarization. Okay, I think this is the last question. Sodium potassium pump exchanges dash potassium in replacement of dash sodium. Option A, 2 potassium in replacement of 2 sodium. Option B, 2 potassium in replacement of 1 sodium. Option C, 3 potassium in replacement of 2 sodium. Option D, 2 potassium in replacement of 3 sodium. Okay, option D, option D, option D. Okay, is everyone answered? Kushbu is saying option C. Okay. Also be careful to look at the ions before the after the fill in the blank. Potassium in replacement of sodium, not sodium in replacement of potassium. Right? So everyone side. I hope everyone answered. Let us see. Okay. Option D is the correct answer. Why? Because question is 2 potassium in replacement of sodium, right? So usually we learned through a sodium pump, 3 sodium plus can go out or in. That is a question. Yeah, again, it's a question. In this case, this is sodium potassium pump, right? Which means they start acting on after hyperpolarization state to bring back the cells to normal back to the resting membrane potential, which is like a higher negative state to a lesser negative state. So which ion goes where? Suppose that is also a question. Yeah, please say. Like you guys can type sodium in or potassium out, whatever. I don't know. Just tell me. So I gave a hint. The sodium potassium pump start acting after the hyperpolarization state. Now just tell me which will go out, which will go in.
Sodium will move out and potassium will move in.
Yeah, sodium will go out and potassium will move in. So we have that figure when he draw in that class. There is a receptor, then sodium to out, 3 sodium to out, 2 potassium to in. So here the question is replacement of potassium with sodium, which means 2 potassium in with respect to 3 sodium out, right? That's why option D. So here it's also written sodium potassium pump system moves sodium and potassium ions against large concentration gradient. So it moves 2 potassium ions into the cell where potassium levels are high, because inside the cell always its higher concentration is potassium. Outside the cell higher concentration is sodium. And the same pump at the same time pumps 3 sodium ions out of the cell, because sodium ions are always at a higher concentration outside the cell and into the extracellular fluid. So that is about all the questions. So we did some demo questions for the week 6. So you guys should, I hope you guys will go and start attempting assignment questions. Then we also see the like last week assignment solution also, right? So you guys solved all the things we were just discussing, right? So if any doubt is there from any of the portions up to now, you guys can ask. So now you guys understood why option C is the wrong option, right? Because 2 potassium in replacement of, if it was sodium in replacement of potassium, it was right? 3 sodium and 2 potassium. But just careful with the fill in the blanks. So first they are asking potassium, which means 2 potassium and 3 sodium. So is there any doubts? Yeah, you can ask or type, whatever the way. What will be the main exam pattern? I think some questions will be like objective type, but some will be subjective. I'm not sure. I will ask the NPTEL team and let you know. Okay. I'm also not sure. I need to ask them.
Ma'am,
Yeah. Do you know?
no, ma'am, it's another question to ask.
Here, okay.
Um, the excitatory signals, they actually
Yeah.
excite the adjacent neurons, right?
Yeah. Yeah. Right.
So what about the inhibitory signals? What will they do?
So inhibitory signal, do you remember when we are discussing last question? In excitatory signaling, they are opening the positive charge ions, right? Ion channels. So inhibitory always open the negative ion channels. So what will happen if negative ion channels are more opening? Will depolarization will occur or not?
That will not occur.
Yeah, which means they are like making it delayed. The process is inhibiting. That's why the name inhibiting.
Okay, so they actually open the negative ion channels.
Yes.
Okay, ma'am.
So any other question? Any other part? So next week, I guess today we discussed the week 5 assignment solution. Next week we'll solve week 4 also. Like next week we'll solve. So week 6 will be also there. We'll adjacently do. We'll do one more session maybe. So we'll try some questions for week 7 first. Then we'll try to solve week 6 question. If the solutions are exchanged with you, then we'll try to solve that. Then we'll try to solve one more session, which will be solution 4. Okay. Because time is already there, 30 more minutes. So this time actually you can ask me questions. So otherwise I can open the question now for the week 4, but the answer is already there. Then we can start discussing also. Either way you want, we'll go ahead with it. Do I need to go and open the week 4 solution? Sorry, assignment solution. Or next week. It's depending on you. Yes or no, please.
We'll do it now, ma'am.
Okay, then let's open. But I don't have the slides prepared for it, so I will open from the tab only, okay? Is it fine?
Yes, ma'am.
So, since week 4. So my screen is not visible, right? Still the slide, Google Slides are presenting.
Yes, ma'am.
Okay, just give me some time. Just opening. Yeah, how's that? Okay, let's see. Now is it visible? Yes, right?
Yes, ma'am.
Yeah, let's go through question by question. Now the answer is actually there. That is the problem. But yeah, we'll discuss. Okay, if you have any doubt, you can utilize this time for asking me. So basically, the week 4 was regarding the, like we were actually discussing the muscle system. Then we started with the structure of neuron, basic structure of neuron. Then ended with the starting what are the, like how neuron looks like, all those things in last 2 lectures. First 3 lectures are basically regarding the muscle system. So that's why the first question is, dash is the smallest functional unit of striated muscle tissue. We discussed it so many times, right? What was A band, Z band? Do you have any doubt in the option? So what was each thing? What is a sarcomere?
The portion from one Z line to the other.
Yeah. So give me 2 minutes. Let's answer, like solve the question like normally we do only. I will remove all the answers, then we'll discuss each question by question. Is it fine?
Yes, ma'am.
Yeah. Yeah. Now I will share. Is it visible?
Yes, ma'am.
So first question, no one have any doubt, right? We all know sarcomere is the smallest functional unit of a striated muscle tissue. Inside that, which consists of actin and myosin and like all these band, right? So if you have any doubt, I will go back to the particular slide and can show also. So just tell me if you have any doubt. We'll go back. Next question is, in the actin myosin complex for muscle function, actin forms dash and myosin forms dash. Option A, thin filament, thin filament. Option B, actin forms thin filament, myosin forms thick filament. Option C, thick filament, thin filament. Option D, thick filament and thick film. What is thick? What is thin? That is the question. Okay, you guys can answer. Option B, Arya is saying. Others, you guys remember this part. I know you guys remember. You guys are smart. Just we'll discuss only. Just guess also. You can guess also. Say any answers. It's fine. Which is thin filament and which is thick filament? Khushbu, Priya, Narendra, Shelke. What is the answer? So, if you guys are not saying, we'll see the solution. So, what is written here? We all know that each sarcomere consists of two separate filaments, right? One is actin and the other is myosin. Myosin filament is actually surrounded by the thin filament. So, myosin have also one more name which is also known as thin filament. Do you remember the structure of the myosin? How it looks like? Okay, let I will share one more thing so that you guys can easily relate to that. One minute. So this was week 4, right? Yeah, week 4. So I'm sharing the screen also. Whenever required, I will show you the figure also. Okay. Now you can see. So in the figure, you can see some sarcomere. Na? This is not the figure. I am asking that blue one is what? Blue color and pink color. Are you able to try to recollect it?
Actin and myosin.
Yeah. What is actin? What is myosin in that?
Pink is actin and blue is myosin.
So in the figure itself, which is thick?
Myosin.
Right. So correct answer is, yeah, you were right. Answer B. Now let's back to question. Yeah, again back to question. Dash leads to actin myosin binding. Dash leads to their detachment. Options are option A, ATP. Detachment is caused by calcium. Option B, ATP and ADP. Option C, calcium ions and ATP. Option D, calcium ions and ADP. What causes actin myosin binding and what causes detachment? Option A, option B, option C, or option D?
Option C.
Okay, Selkie is saying option A. Okay. Ari is saying option C. Okay. Kushbu is saying option C. Okay, you guys can unmute and tell me why you guys are saying the respective option. Then we'll discuss. First question, dash leads to the actin myosin binding. What is the first process that activates that? What is how they are getting the information to how this is the time for binding, right?
The unmasking of
Yeah.
actin sites.
Uh,
That is done with the help of calcium, which binds to the troponin and unwinds it.
yeah, correct. Then why they are saying dash leads to their detachment? Do you,
ATP leads to the detachment.
so what will happen in that process? How ATP is making the change? So whenever that is the time for ATP will go and bind at the correct responding site in the troponin part. Then at that time they will know. So that time it will trigger the detachment, right? So So correct option is option C only. So you can read it also. When calcium ions bind to troponin, the shape of troponin changes, which is the first process. So these calcium ions are basically released by the sarcoplasmic reticulum. How sarcoplasmic reticulum to know that? Because again in the back there are some process of action potential happening, which neuron comes to play a role. Then again central nervous system comes to play a role, right? So after all this process, Ca2 plus will release, which will cause the binding of that ions with the particular troponin. Then what will happen? This moves the tropomyosin from the myosin binding site on the actin filament, right? So it is basically unblocking them. The unblocking is basically providing the opportunity for the myosin heads to come and bind to the actin filament, right? Then it will slide. Do you remember the sliding filament theory? So basically then that occur. So whenever that process is sliding occur, after that ATP is the one to initiate the process of detachment. So whenever ATP binds, what will happen? Myosin head start detaching, which means tropomyosin will, which allows the tropomyosin to reattach to the active site on actin. Without ATP, myosin heads cannot detach. So always detachment is initiated by ATP, and like the all the process of attachment or the sliding is initiated by the Ca2 plus release. So among the option, most suitable option is option C. Next question. Which of the areas highlight a region that has myosin but no actin filament? Option A band. Option B, A band, exon. Option C, exon. Option D, A band, I band.
Headstone.
Sorry, I didn't.
Headstone.
Okay. Any other option? Option C, option C, option C. Oh, everyone is saying. Good. So let us see the answer. So what was the answer? H1, right? Why H1? Do you remember? Just now I showed that figure, right? So whenever in the release state, what is H zone? It contain only just the myosin. So if you are going and check back on week 3, you can find the proper structure of a sarcomere unit. So please, if anyone have doubt, please go and refer to that slides. Okay. Then next, which of the area contains an overlap of actin myosin? Now it's overlap, right? A band, AB band, exon, AI band.
A band.
Okay, Arya is saying A. Other options? Selke is also saying, Kushbu is saying A. Okay. Is everyone is sure? Okay, let us see what is the answer. So just try to recollect the complete image of sarcomere in mind so that it will be easy. It will be helpful for your exam also. Yeah. So what is A band basically? You guys told me A band, right? So A band actually only contains which element? Thick element or the myosin element. But anisotropic band contains both thick and thin filament. So do I need to open the figure again? Not that one, the proper unit. If I need to, I can go and again show you the image. Okay, let us share. So So I'm not going to the slide, but I will show you an image, okay? I hope this is visible. Look at the image. You can see, right? A band is the central part, which consists of only of thick filament or the myosin filament. This is the overlapping part, right? So which is the correct answer?
AI zone.
Yeah. So again, I need to share. Okay. That's why they have written there anisotropic band contains both thick and thin filament. Length tension relationship relate the strength of and dash to the length of muscle at which contraction occurs. Sorry. Now, isobaric contraction, isometric contraction, hypobaric contraction, and hypoxic contraction. It is related to pressure. I don't know. You guys just tell me. Tension. Even the word is there. Tension. So something related to tension is always what? If you are unable to recollect also, it's fine. Just tell me. Just some guesses. Only four more questions. Then we'll end the session.
Is it isobaric?
Okay. Isobaric. One option is option B. Okay. Is any other option? Okay, let us see what is the answer. So muscle is always, this can be variable. There are two variables, right? Length and tension are there. So when something is described as isometric when if the muscle tension changes but the muscle length remain the same. So what was the question? Length tension relationship relate the strength of sun dash to the length of muscle at which the contraction occurs. So what is the answer here? Huh? It is isometric. Did you got it?
Yes, ma'am.
Yeah. Okay, 1 minute. What answer did I set for this question?
AI band.
No, it's not AI band. So the question is basically overlap region, right? Sorry, contains an overlap of actin and myosin. Okay, so during the contraction, right? The same figure that I have showed was the resting figure, right? That time it contained only the myosin. If it's the contraction is occurring, is that central part also covers or not? Which means the correct answer is A band only. You were right. Okay. So fifth question, correct answer is A band. So sorry for the confusion. Just think in that way. The contains an overlap of actin and myosin. So basically in all contractions, actin is coming, right? Sliding. So that sliding over happens in that part only. So basically the answer is 5A. And sixth we already discussed, it's isometric. Question 7. The muscle operates at the greatest active tension at the muscle's optimal length. The optimal length is also called. This we have discussed, like we have learned. Shortest length, maximum length, resting length, running length. There is one more name for that we were discussing, and also I showed that in the week 6 slide also it was there. Which is the option? Anyone?
Resting length.
Yeah. Option B. Okay, option B is maximum length. Okay. Two people are saying option B. One is saying option C. Is there any other option? Correct answer is option C. Why? Because in the question itself they are saying, right, muscles optimal length. Do you remember muscles optimal length? Then again after it's again increasing the length, what will happen? The process is going back. That's why I showed that contraction image, right? So just go back and refer to that. So always greatest active tension is happening at the muscles optimal length, and that length is also called as resting length. Okay, so option C. Then the subunit of troponin that has a higher affinity for tropomyosin. It's tropomyosin, okay? Troponin I, troponin T, troponin C. This is basically like some extra syllabus question. There are 3 subunits in troponin, and which of the subunit has greater affinity for tropomyosin? Just guess. It's fine if,
Troponin T.
okay. Kushbu is also saying troponin T. Sure? Okay, let's see. What is the correct answer? Troponin T binds to the complex to tropomyosin. What is C? See the name itself, troponin C, which means that means calcium. And I, what will I do? So basically, T binds with the tropomyosin, and I is basically doing the interaction of the complex with the total tropomyosin. So that's why these 3. So troponin C binds to calcium, troponin T binds to tropomyosin, and troponin I basically do the overall checking of the interaction of these 2 filaments, which are troponin and tropomyosin. So I hope the answer is clear. Next question is, intrafusal fiber is dash, that is also a dash. Neuronal type, chemosensor. Neuronal type, mechanosensor. Muscle type, chemosensor. Muscle type, mechanosensor. Intrafusal fiber. We know this thing very much when we were learning this, right? What kind of sensor is that? And what type of fiber is that? Is it muscle or is it a neuron?
Muscle.
Okay, then what is the sensor?
Mechanosensor.
Okay, so you are saying option D. Option D, 2 people said option D, right? Okay, let us see. Basically, correct answer is yeah, it's definitely a muscle fiber. Then what is the specialty of these detectors? What can they detect? They can detect the amount and rate of change in length of a muscle. They constitute the muscle spindle and are innervated by both sensory and motor fibers. These fibers have a mechanosensory capacity, forming part of the monosynaptic stretch reflexor. So basically the answer is they are muscle fibers and they are responsible for proprioceptive function, and they are what in nature? Mechanosensory in nature. So option is option D. 9. right? Now the last question. Last question is myotubes are mononucleated, multinucleated, non-striated, non-contractile. Easiest question. Yeah, this is easy, I guess, if you all remember.
Multinucleated.
Okay. Any other option? Do you remember how the each separate joins together to form something? Then only the wall is there. Then they will start forming a structure which consists of so many lunged tube with it looks like a pipe, right? So basically answer is option B, right? So that is about all the four questions. Sometime we'll go back and refer to third, second, first also, okay? So that you guys will remember the session also and it will be helpful for your exam also. So that is about today. Hope to meet you on Saturday with discussion of week 7. Then we'll try addressing week 6. Then we'll go and check week 3 also. Okay? Is it fine?
Fine, ma'am.
I am sorry about one question confusion, that in between question. Okay, bye. See you in next class. Thank you.
Bye, ma'am.